Thanks very much, Julie. And we probably not. I think I'll stay here, especially since we've got a group. And thank you to Heather and the organizing committee for putting together this really timely and important forum. It's exciting to see a room filled with people that are concerned about these issues that Peter Ross so eloquently raised. And Peter did a great job of framing the scope of the issue, and that allows us to kind of launch right into talking about the work that we're doing locally, kind of in the Salish Sea region, on plastics. So to start off, I just wanted to frame a little bit our approach to trying to generate a better understanding of some of the impacts and the pervasiveness of plastic-related issues in the region. So we're using a focal species approach. So we're trying to work at multiple trophic levels. So if you think about food webs, trophic levels are the different levels at which organisms feed, from primary producers up to top-level consumers, and use a suite of species that we've selected that give us insights and tell us something about the actual phenomenon. So a biological indicator has to be sensitive to the issue of interest. And that, for us, is the distribution of plastics and the consequences of interacting with those plastics for populations. Secondly, use bioindicators to monitor trends in marine and freshwater plastics over time. Use a food web approach to develop models of distribution and relative abundance in marine ecosystems. And finally, assess impacts of plastic exposure on marine wildlife. So basically, a lot of the research to date has been descriptive in nature, and that's of necessity. We need to understand the scope of the problem. That, as Peter really nicely summarized, has been pretty well documented. You know, we understand that plastic is pervasive in marine food webs, as well as freshwater systems. And now we need to get to the so what question. So science tends to proceed from description to moving from the pattern to try to understand the mechanisms. And Peter, again, is right at the cutting edge of that sort of work. I also want to mention that all of this work is done actively, and I think it's represented really nicely with Patrick and Robyn here, who are going to speak as part of my comments. This is work that's in many respects led by the undergraduate students at UPS. They come to me with fantastic ideas, and a lot of the work that I'm describing here is not only done by them, but it's been conceptualized by them. So I wound up kind of inadvertently in the world of plastics-related research. I'm a seabird biologist and conservation biologist, and one of the growing kind of recognized threats in the world of seabirds has been the impacts of plastics. Many of us have seen pictures and understand, you know, the kind of the iconic issues associated with albatrosses in the Northwest Hawaiian Islands. And I work on some similar species, and it was kind of through that perspective as a potential impact on seabirds that brought me into the world of plastics and since has led us to diversify. So a couple of the species that we work with, northern fulmars and sooty shearwaters, are up on the screen. They do ingest plastic. Here are a couple of samples of plastic from individual birds. These are samples from individual shearwaters, sooty shearwaters, and northern fulmars. In terms of plastic ingestion, we find significant levels of plastics in these birds. So northern fulmars are on the right there. They have upwards of 90% of all individuals sampled have plastic in their guts. Sooty shearwaters, it's a slightly less bad story, but it's still well over 50% of all birds have plastic. And if we're thinking about this the concept of biological indicators, they need to be sensitive. So they need to reflect relative abundances or accessibility of the plastic in the system. And so results such as we've just got on the screen here, which show significant differences in the ingestion rates of plastic for birds in Alaska versus California and Washington, suggests that that might be true. So these birds are reflecting on some level the abundance of plastic in the systems that they inhabit. So we're also trying to move beyond, again, as I mentioned in my initial comments, beyond this descriptive and move towards really the so what, the consequences. And I have a recently graduated student who's trying to do some work along these lines. So we did a proof of concept study. This is just to illustrate a direction that we're trying to take in the lab. This is still early days for us, but using common MERS, which are a seabird up on the screen, and understand PBDE concentrations in liver versus feather, such that we might be able to use a less invasive technique. As those of you thinking, liver, sampling liver is usually not so good for the health of the organism being sampled. So can we use develop less invasive techniques to sample and understand chemical signatures through feathers? And by doing that, track trends over time. So we don't have any results from that. This was really a proof of concept study. But by working on seabirds, it kind of led us to think about, well, seabirds can ingest plastic two ways. One, they can ingest it directly, mistaking it for food, or they could ingest it inadvertently by consuming things that have eaten plastic. And so that led us into studies into plastic ingestion by forage fish. And that work was led initially by Bonnie Worth, shown here, and a few pictures of fibers and plastics that she found in forage fish. So she looked at a suite of species. Julie mentioned that we work on rhinoceros auklets. We actually used diet samples from rhinoceros auklets, which are bill loaders, so they're like puffins. They'll bring fish back into the colonies. If we collect those samples, we can then use those fish for multiple research questions, including plastic ingestion by those fish. And you can see every species that we've looked at so far has evidence of plastic ingestion, from as low as about 1 in 10 individuals to upwards of 50% in some species. And as Peter mentioned, with the water samples, what we find in fish is consistent. The vast majority of the plastic that they ingest is comprised of filaments, with a much smaller fraction comprised of pellets and fragments. We did a study here, and those of you who are scientists in the room, you're like, interesting, 2 points. You can always draw a straight line and make a relationship between 2 points. We're working on adding another year to see if this relationship holds up, but it is a concerning one, at least initially. So this is the frequency of occurrence of plastic in multiple fish species in 2008, and then those collected in 2013. And this is suggestive of a doubling in plastic ingestion. Again, we need more data, but it's certainly consistent with plastic production trends. And with that, Robyn Thomas will speak on some of the research that she's been doing for her senior thesis, kind of building on some of those initial findings of plastic ingestion in forage fish.
All right, thank you, Peter. I'll going to be talking about what I've been up to this past summer. I was looking at the gastrointestinal tracts of 3 different forage fish species, which I'll talk about in a second, from multiple locations in the Puget Sound. To reemphasize what both Peters were saying before, basically all the plastic that I was finding in these fish were fibers. You can kind of see on the screen there, they look like they're little hairs, and that was basically all that I was finding. I looked at about 140 fish, and only, I had like 350 pieces of plastic total, and only one of those pieces was not a filament. So that was pretty, not really surprising, but interesting. Here are some of my results from this past summer. As you can see, I had 6 different sample sites. So over in Hood Canal, if you're not familiar with the Puget Sound, it's the gray dot on the screen. And Dabob Bay had the highest instance of plastic ingestion from forage fish. And I had hake forage fish, which are Pacific hake, and those are, I didn't have like multiple species for each site, but I still think that this is pretty demonstrative of the plastic ingestion that we have here in the Puget Sound. Down in Nisqually, down by Olympia, we had the lowest frequency of plastic ingestion in forage fish, but still I think across the board we had pretty significant plastic ingestion. And with that, I will pass off to Patrick Wallin, who looked at benthic fish plastic ingestion.
All right, thanks. So what I did is I looked at benthic organisms, those plastics organisms at the bottom of the ocean, and I did this because there are a lot of tests that have, a lot of data that's been saying that plastics eventually sink down to the bottom. This is due to fouling because of algae that attach to it, so it's not super scientific, but it gets denser and eventually will settle on the bottom. So I looked at 2 different species. I looked at the English sole, which is the fish on your left, and that's a flat fish. It's actually commercially fished. And I looked at the spotted ratfish, which is the other fish. That one lives a little, it doesn't live, sit on the bottom like the sole does, but it does swim around on the bottom and considered a benthic fish. So the Department of Fish and Wildlife captured, took trawls of these fish at 9 different sites across the Puget Sound or across the Salish Sea. And so what I found was that the frequency, so this graph is the frequency of occurrence of microplastics in these fish. And I did it at 5 different sites. I had 5 different comparisons. And you can see they're pretty high. There are 3 of the English sole sites had 100% frequency of occurrence, which is really high, obviously. And then, so spotted ratfish was slightly lower, total average of occurrence, but it was still almost just as high across the board as the English sole. So then I compared this to the data that Robyn got. This is frequency of occurrence. So the benthic fish are in red, pelagic are in blue. And you can see it's pretty similar throughout the watershed. There wasn't any significance, but you can, I think it's really important to realize that this isn't just a surface level problem, that benthic organisms are also affected by plastics. So I'll hand it back to Peter.
Thanks, Robyn and Patrick. And you can see that this is work, again, that was undertaken completely by Robyn and Patrick. And it's exciting work, and especially the work with that Patrick has done looking at benthic species is an area of this 3-dimensional environment that fish and birds and marine mammals inhabit that's relatively poorly studied. So moving that part of our understanding forward is really important, because as Patrick mentioned, a significant amount of plastic settles out of the system. And it's easy to think, oh, it's not in the water column, we don't need to worry about that, but everything that's down at the bottom or foraging on bottom-dwelling species is going to be engaging with it. So we've also tried to move down even further trophically into filter feeders. And this is work that was, again, initiated and really conceptualized by students in my lab, including Nick, Olivia, and Eliza. So using mussels in this case, here are images from microscopic images of some of the filaments found in mussels. A quick summary slide here. This represents microplastic ingestion by mussels, the mean number of plastic particles, the number of locations. And this is just like the previous 2 studies, Robyn's and Patrick's. This is work that was done in collaboration with Washington Department of Fish and Wildlife and their Mussel Watch program. So we have samples from them and are looking at plastic ingestion or the presence of plastic in mussel samples from throughout, as you can see, really good representation of Puget Sound right up to the Canadian border. Other work shows that as mussels get larger, which means that they get older, they tend to inject, they have higher plastic loads in them, so that they do tend to accumulate plastic over time. These are from 2 locations, Destruction Island on the outer coast with the red squares, and then Key Peninsula with the blue diamonds. So again, trying to move beyond just this descriptive, we've also had students like Kieran and Olivia working in controlled experimental studies. And again, these are proof of concept studies, but I think they point the way to directions that we're trying to take our research in the future. So in particular, Kieran's work, where she raised Medaka fish, shown here in that picture, in different plastic environments, including a control, clean plastic and then plastic that was laced with PCBs to see if that PCB was being transferred to tissue in the fish. So this is, again, these are early stages of this longer term work, but it's an approach that we're trying to get at the mechanisms and the impacts. And then just to wrap up, another student came in and said, well, I'm really interested in the plastics work, but I'm a kayaker, I'm a freshwater person, I don't really think about the ocean. Can we work on freshwater plastics? And I said, okay, sure. Don't know much about that. You know, we know about the plastic part, but I assumed that plastic was actually reasonably well studied in freshwater systems, and it turns out to be incredibly poorly studied. And so that opened up kind of a new research opportunity, and Carson, again, who's shown here, led that effort, and we're trying to develop this direction of research further as well, where she looked at fish in the Puyallup and the Green Rivers and did a comparative study of coho salmon, sculpin species, and then dace species, and found, and again, the prediction was that because of the different life histories of these fish, we would see different plastic ingestion rates and or loads. And that's actually what we found. So coho, being kind of up in the water column more than the dace and the sculpin, had the lowest plastic loads of the three. But again, this is just an initial study, kind of exciting results. It points to, again, this idea of using species selectively as indicators. And again, that comes back to kind of one of our central goals with the research. So we'll finish there. I'm not sure if we've got time for a question or two. Okay. Yes?
You definitely got, studies on what's happening in the water and what's happening in the fish. Is anyone doing studies on their waste or elimination, like how much passes through, like
that's something that needs to be done. Peter, I don't know if you're aware of, I know it's certainly of interest because as Peter mentioned before, the retention time is fundamentally important. You know, as I think his example of a microplastic going through a humpback whale is going to be inconsequential to that whale. So it's a scale issue. But I don't know that literature, Peter. Do you have a response to that?
There are a couple studies, but they're very early stage, so a lot more needs to be done.
Yeah, but you've identified one of the key areas is that retention time. The longer that plastic persists in an organism, whether it's a zooplankton or a seabird, the longer the time that those chemical contaminants can leach out or desorb from the plastic and enter the tissues of the organism. Yes, sir.
Are your students getting published?
Yes, we have a paper in Marine Pollution Bulletin that's coming out, and Patrick and Robyn, I've talked with them, and the idea is that their senior theses are going to be turned into publishable papers. So, yeah, we feel it's really important it's publishable quality work. They're really good. It's important work, and so we're trying to get it out there into the literature. Yeah.